in including Institut Gustave Roussy in France and the Polish Academy of Science in Poland, but different other institutions in Africa. So the main goal of Permagena is really to assess the readiness level of personalized medicine implementation and to reinforce precision medicine capacities in the region. And to achieve this goal, so we have 4 main specific goals or work packages. The first work package is about the situation analysis, the situation analysis and an inventory of existing precision medicine. So in this work package, we are working on looking what is existing and what is missing to implement precision medicine in the North African region. And the work has been done, and we hope that we will present the results of this work package during our final meeting of the project that will take place in around 2 months. The second work package of Permagena is about a pilot project on precision oncology. So we decided to work on lung cancer as an example to start implementing a precision medicine for different other diseases. So this work package, we are generating genomics and transcriptomics data to identify the tumoral and the genetic profile of this type of the disease, but also to assess the whole process for the implementation of precision medicine going from the gene to the therapy. Then we have a third work package, which is more on training, dissemination, and communication. And finally, we have a work package that is more dedicated to policymakers. And the main goal of this work package is to elaborate a kind of a roadmap and recommendations for health authorities on how we can really implement all together with all the stakeholders the precision medicine in North Africa. Along with all these work packages, so in Permagena, we have 2 additional task forces. So the first task force is about the implementation of molecular tumor boards. And we all know the importance of these MTB in precision medicine. In French, it's called RCP moléculaire. And we are doing this for lung, breast, and ovarian cancer. So we put in place a kind of a very multidisciplinary team, including oncologists, medical geneticists, pathologists, molecular biologists, and bioinformaticians to work together on the implementation of different genetic testing. Those specialists are coming from different institutions. And here we are really very glad to collaborate with Gustave Roussy Institute and with our partner, Dr. Etienne Roulou, who is really helping us a lot to put in place those tumor meeting boards. For the genetic testing that we are performing in this task force, so we started, as I said, with the breast and ovarian cancer for the BRCA1 and BRCA2 genes, and we put in place this testing at Institut Pasteur of Tunis. We also used another panel for a multi-cancer panel to really try to identify mutations associated with different cancer types. So we have around 40 genes that are tested for patients. And here we are talking about genetic testing. So this team that is really formed by different specialists, so they really meet to discuss about the genetic results and to correlate the genetic profile with the clinical data. And the main goal is really to deliver a kind of a clinical report that will help oncologists to decide about the right treatment that can be given to the patient based on his genetic and molecular profile. But as you know, when we make a kind of a sequencing for some patients, so we are generating different types of mutations. We have those reportable mutations that we can put in the clinical report and that can help the patient to get his treatment. But also we have what we call the non-reportable results. Non-reportable results, this includes novel mutations or mutations that are classified like variant of unknown significance. So we decided to make kind of further investigations to validate the functional impact of the identified variants. And to do so, we put in place a second task force for functional genomics. And I think that for this webinar today, we will be discussing this, this topic. For the second task force, so it's led by our colleagues, Dr. Hiroki Taniguchi and my colleague, Dr. Ines Bini, with different other PhD students and postdocs that are really working to put in place a CRISPR-Cas and the technology of CRISPR-Cas to validate the functional impact of the variations that we are identified, and to do different other functional genomics assay to really assess the biological effect of the variants that we are identifying. So the work that we are doing in Permagena has two different aspects. So we have the research part on the specific task force to validate the functional impact of different variations, but also we have the medical and the clinical part on patients to deliver and to put in place genetic testing and to deliver clinical reports for patients in order to give them the right treatment for their diseases. And so that's it about PERMAGENA. PERMAGENA is a project that created the appropriate environment for implementing precision medicine in North Africa. It's a project that during this project, we build a new strong partnership with at the national level, regional, as it's a project that covers 3 countries in the region, and also at the international level. So we build a really strong partnership with different institutions. Also, the PERMAGENA project helped a lot to start working on national strategies for human genome projects and for precision medicine implementation in the region. Because we are working on the recommendations and the roadmap, so we are trying to help authorities to see how to ensure the translation of knowledge into practices, health, products, and services to the benefit of our population. And all this really needs political willingness and efficient plans and strategies for cost-effective solutions adapted to African socioeconomic settings. So this is what we are trying to do in PERMAGENA. And I hope that during this webinar, we will have the opportunity to talk more on this project. And so thank you very much. And I will give the floor to my colleague, Erika Sella, to present about the EU-Africa PERMEL project. Thank you.
Thank you, George. Thank you very much. Good morning. I hope you can hear me. My name is Erika Sella. I am from Spain. I work in Innovatec in Spain, but I am here as coordinator of the EU-Africa PERMED project. We are very happy to collaborate with PERMAGENA to organize this type of webinars. So doing things together I think is a very good thing when we are both trying to reach the same objective. So I would like to share my screen with a short presentation of the project. It's a short presentation because we don't have too much time. I'm sure many of you know about EU-Africa PERMED because from the participants, I see some names that are familiar to me. But in any case, I will give you a very short presentation of the EU-Africa PERMED, building links between Europe and Africa in personalized medicine. This is a coordinating, and I will put it in presentation mode. Okay, this is a coordinating and support action funded by the Horizon 2020 program from the European Commission. And the main goal of this project is really to facilitate and strengthen the research and innovation collaboration between Europe and Africa in personalized medicine. The idea is to foster joint projects and programs. And a very important objective of the project is facilitating the participation of African organizations in the International Consortium for Personalized Medicine. I'm not going to go into details about IC PERMED because we don't have time. There is information in the website, but this is really a platform that was launched by the European Commission and the European member states to work together towards facilitating the implementation of personalized medicine. It was started in Europe, but it's really opening up to other organizations from across the countries. So EU-Africa PERMED is a consortium of 13 partners. We have 6 partners from Europe and 7 partners for Africa. You see here the organizations. And this is a picture of the consortium in Cape Town. We have the Ministry of Health from Italy. We have Maria Jose here with us. She's representing the Ministry of Health of Italy, a partner in the project. We also have the European Clinical Research Infrastructure Network, ECRIN, based in France. This is a pan-European infrastructure. We have INSERM from France also, and ANR, the National French Research Agency. And we have the Instituto de Salud Carlos III. It's the National Health Institute of Charles III, also based in Spain, and Innovatec. But from Africa, we have several organizations from different regions. From South Africa, we have the South African Medical Research Council. From East Africa, we have the African Population and Health Research Center, and NACOSTI, the National Commission for Science and Technology. From Egypt, we have another organization, ETHIT, the Egyptian Center for Innovation and Technology Development. And from the West, we have IRESEF, which is the Institute for Health Research, Epidemiological Surveillance and Training. Very quickly, these are the objectives of EU-Africa PERMED that are translated into different work packages. First of all, we did a mapping of to know the situation of the personalized medicine in Africa, or both scientific and policy context. We have another work package working on cross-border collaboration, is how to explore and analyze ways of collaborating, which are the areas where collaborating between Africa and Europe is of value for both regions, and preparing the action plan. Then we have another important objective, which is how to translate the message to policymakers, because it's good to work and engage researchers in collaborating together, but funding is always important and needed. So we also need to convince the people at the policy level that personalized medicine is important, and collaborating between Africa and Europe is something that should be included in the policy agenda. And also the integration, as I mentioned before, the integration of African countries in ICPERMED. The objective number 5 is capacity building, which is to carry out capacity building and training activities addressing relevant issues for Africa. And this webinar is precisely the type of activities we are doing in capacity building, and very nice to collaborate with other projects such as PERMEDINA. And we also have an important objective, which is dissemination and communication, to translate the findings from our project and all the results to all relevant stakeholders and also to the wider audience. Very quickly, these are some of our main results so far. In terms of incorporating African organizations in the ICPERMED consortium, we are happy to say that now we have the South African Medical Research Council and the Egyptian Academy of Scientific Research and Technology as full members, and Tunisia is in the process of becoming now a full member, represented by the Ministry of Health and the Ministry of Science and Technology. So we have been able to incorporate 3 African organizations in ICPERMED, and I think that's a very big success to incorporate the views of Africa in these international platforms discussing about how to advance the implementation of personalized medicine. South Africa also joined one of the IRA-PERMED joint transnational calls. These are joint funding activities promoted by the European Commission to support international projects, and South Africa was able to gain one of these projects that was actually coordinated by a South African researcher. We are also one of the projects that has been selected as to contribute to the European Union, African Union Innovation Agenda implementation. As I told you, it's very important that personalized medicine and the collaboration between Africa and Europe is included also in the high-level policy dialogue that is taking place between Africa and Europe, because if it's included, then there will be funding available in the programs, because policy decisions are normally the ones that help to implement topics and to implement calls related to this activity. Another important output of our project is the EU-Africa PERMED Action Plan. This was recently launched in a meeting we had in Senegal, in Dakar, in June, and it provides some recommendations for collaborative actions responding to common health challenges and needs identified through exchanges with African and European stakeholders. This is a very important document because we are presenting a set of concrete and well-defined actions that can be taken up by already existing programs and such as, for example, the European Commission Horizon Europe or the EDCTP programs that are already established and that are funding health research activities between Africa and Europe. And we are recommending specific actions that they could implement if they want to support the collaboration in personalized medicine. And we are also supporting the setting up at regional level of personalized medicine groups in the different African regions because we feel that this is also a way to advancing the personalized medicine agenda in Africa. We've seen, based on the mapping results, that Africa is a very big continent and the situation is not the same in the different regions and the different countries. There are different levels of maturity, so the activities that need to be done to advance personalized medicine have to be adapted to the social and economic situation of the countries. And very finally, this is also something very recent, is the Dakar Declaration for a Stronger Collaboration between Africa and Europe in Personalized Medicine. I will not read it, but I invite you all to enter into our website and have a look at this declaration that came up from the last stakeholder workshop in Dakar, and it summarizes a shared message from the project and also the stakeholders that participated, highlighting the importance of international collaboration for advancing personalized medicine, and presents key points for policy action considered to be relevant for a stronger and more sustainable collaboration between Africa and Europe in the field of personalized medicine research, innovation, and implementation. It's really a document that could be used by researchers and stakeholders to advance the personalized medicine agenda in their countries. We know that, for example, our partners in Senegal will present this Dakar Declaration to their Ministry of Health, so that personalized medicine research and innovation is taken up as a key opportunity for advancing the health of the population in the country. So please go to our website, and if you want, read the Dakar Declaration, because we feel that it really summarizes what we want to come up out of this project. Nothing more. This, you can follow us in our website, also in Twitter and LinkedIn. And very happy to be here if there are any questions, and to join this very interesting webinar, that I feel happy that we are also reaching far countries such as Japan. So we are not only now collaborating between Africa and Europe, but we also reaching out to faraway countries such as Japan. So very happy to be here with you, and thank you very much for your attention.
Thank you very much, Erika. Thank you for this great presentation of the EU Africa PerMed project, and I'm so happy to see all what you did, and also the collaboration that we are putting in place between PerMedine and the EU Africa PerMed, and the potential outcomes that will come from these two projects, mainly in the involvement of the IC PerMed consortium. And I have to also thank my colleague, Dr. Hiroki Taniguchi, who is from Japan, and who is the one who was able to bring some partners from Japan, such as our guest speaker today. So I will give the floor to Dr. Hiroki.
Okay, so it seems like the time is running, so we want to keep some time for the presentation. So I'm going to go with the introduction of Professor Akihiro Fujimoto. He's a professor of Department of Human Genetics, graduate School of Medicine, the University of Tokyo. He received his PhD from University of Tokyo in 2008, and then joined a very famous research institute in Japan, Riken Institute, and he has worked on the Japanese whole genome and liver cancer genome as a first time. And his current interest identification of genetic variation and somatic mutation, and have a better understanding of their functional impacts. And actually, the floor is yours, Professor Fujimoto. Could you talk about today's presentation of yours? Thank you.
Okay, so Dr. Taniguchi, can I share my screen?
No, I think you have to get permission from the organizer to upload the oh, now, yeah, it's there.
Yeah, no, I think I can share my screen. Okay, and thank you very much for the very kind introduction, and thank you very much for giving me opportunity to talk about my research. It is a great honor to be here. So today, I'm going to talk about the whole genome sequence using short read and long read sequencing technology. So first, I would like to explain my previous study. So as Dr. Taniguchi mentioned, I performed the whole genome sequence of a Japanese individual in 2009. That's a very long time ago. So at that time, we developed a method to identify genetic variation and identify the structural variation from a single Japanese individual. Then I moved to the analysis of whole genome sequence of liver cancers and developed a method to detect somatic mutations and identified driver genes in liver cancers, and I identified integration of hepatitis B virus into the human reference genome. Unfortunately, these papers are published in Nature Genetics and Nature Communications. So, and I also joined the International Cancer Genome Consortium and analyzed and worked for the pan-cancer analysis of the International Cancer Genome Consortium. I developed a method to detect microsatellite mutations, and I also detected a method to detect the driver gene considering the 3D protein structures. So these papers have been published in about 4 years ago and 8 years ago. So but I noticed that there are some problems in previous my previous studies. So our study identified a lot of genetic variation and identified a lot of somatic mutations. However, I thought that there may be a lot of genetic variations unidentified by the next generation sequencing technologies because they are the lead length of the next generation sequence technology is very short, and the analysis method is incomplete. And I also consider that our previous study did not analyze the full-length transcript. I think that analysis of full-length transcript is very important to estimate the function of the proteins. So I consider that our understanding of human genome is still incomplete. So about 10 years ago, when I started the analysis of the next generation sequencing data, I thought that we obtained a complete map of human genetic variation, somatic mutations. But actually, that's not correct. So there are a lot of holes in understanding of genetic variation, somatic mutations. So I decided to start the next study to identify previously unidentified genetic variation and transcript. So for this purpose, so we started the analysis of the long read sequencing, and we started the development of the analysis method of short read sequencing technology. So this is the limitation of short read sequencing technology. The short read sequencing technology is difficult to identify the repeat-mediated structural variations. If the breakpoint of structural variation are in the repeat sequence, the short read sequencing technology cannot identify the breakpoint. And second problem is identification of inserted sequencing. So if we use some good analysis method, we can identify the presence of insertions using the data from the next generation sequencing technologies. So we can know that there is insertion here. However, the insertion is longer than the read length. We cannot observe the inserted sequence. And third problem is full-length transcript is very difficult to be observed directly. So short read sequencing technology can estimate the expression level of gene. However, the connection of exon is very difficult to be estimated. So we started the analysis of long read sequencing technology. So today I would like to talk about two topics about short read sequencing technology and 2 topics about long-read sequencing technology. First, I would like to talk about a comprehensive analysis of indels in whole genome microsatellite regions and microsatellite instability across 21 cancer types. This study was done as a part of ICGC, pan-cancer project. Then I would like to talk about systematic analysis of mutation distribution in 3D protein structures. So we consider that the analysis of 3D protein structure can identify novel driver genes. So I think this is a very good method. And then I would like to talk about the whole genome sequence and transcriptome sequencing using long-read sequencing technology. So first, I would like to explain microsatellite cancer. Microsatellite is defined as a tract of repetitive DNA motif composed of short repeat unit. The mutation rate of microsatellite has been known to be higher than other genomic regions because of DNA polymerase slippage during DNA replication. So due to this fragility, the microsatellites are used as a marker of genomic instability in cancer. So microsatellite instability, MSI, has been used for microsatellite molecular diagnosis of Lynch syndrome and cancer with mismatch repair deficiency. Now it is known that MSI phenotype are generally harbored with higher burden somatic mutations. And MSI can be also used as a marker to predict the efficiency in immune therapy. So to reveal the whole genome mutational landscape of microsatellite regions, we analyzed indels in microsatellite region for the 2,913 cancer genome sequence gathered by International Cancer Genome Consortium. So first, we defined the region to analyze. And the definition of microsatellite is not very simple. So we used the microsatellite region using 3 previously published methods. So using this method, we identified more than 9 million microsatellite regions in the human genome. Then we generated the method to identify mutations in microsatellite. So microsatellite regions by 3 softwares, and we gathered the leads that cover the microsatellite region. Then we count the number of lead, a number of repeat, and included in each repeat. Then we determine the probability of the somatic mutation using this likelihood function. So using this function, we identify the somatic indels from each microsatellite. So then we estimate the sensitivity and the specificity of this method. So we compared our result with somatic indel calls from the 4 International Cancer Genome Consortium pan-cancer indel callers. As you can see here, in the microsatellite regions, so our method identified quite a larger number of insertion deletions in microsatellite region compared to other insertion deletion caller provided by International Cancer Genome Consortium. So this result strongly suggested that our method is very, very effective to identify insertion deletion in microsatellite regions. So we also compared the number of microsatellites between our method and the insertion deletion codes detected by PCOG, International Cancer Genome Consortium. These 2 methods are very, very highly correlated. So this result also suggests that the reliability of our method. So we also performed the experimental validations using capillary electrophoresis and estimated false positive rate. The false positive rate was estimated to be 7% in our method. So this result suggests that our method is reliable. So we analyzed the whole genome sequence data of about 3,000 pan-cancer samples. So, and this is a mutation rate of microsatellite. So but the majority of the microsatellite didn't have any mutation. So we focused on the microsatellite with somatic mutations. And the pattern of the error rate are quite different among the pattern of the microsatellite. And 80 microsatellite has higher mutation rate compared to other microsatellite. Then we compared the mutation rate of the microsatellite. So this is the graph of the mutation rate. So the x-axis shows the type of cancers, and the y-axis is mutation rate of microsatellite. As you can see here, several samples have higher microsatellite mutation rate compared to other samples. And colorectal, as expected, colorectal sample and somatic colorectal cancer, somatic cancer, stomach cancer, and uterus cancer has the sample with very high mutation rate in microsatellite regions. These two graphs show the mutation rate of the SNV and somatic indels. The mutation rate of somatic indels are highly correlated with the mutation rate of the microsatellite. So we also estimate the pattern of mutation in microsatellite regions. And in the MSI and MSS samples, the deletion are more predominant compared to the insertions. So then we analyze the factors that affect the mutation rate of microsatellite. So we analyze several factors, such as the epigenetic factor, DNA structure, and sequence motif. Among them, the replication timing and microsatellite mutation rate are highly correlated. So this result strongly suggested that the replication timing affects the mutation rate of microsatellite. And interestingly, this pattern is quite different and completely opposite in the MSI samples and MSI deletion in MSI samples and deletion in MSS samples. So next, we also analyze another factor that can affect the mutation rate of the microsatellite. We found that the shape of DNA is significantly associated with the mutation rate of the microsatellite. As you can see here, the DNA shapes, this is the angle of DNA base, is significantly associated with the mutation rate of microsatellite. So we also identified the 20 highly mutated microsatellites. So we performed the genotyping of these highly mutated microsatellites and found that these 20 highly mutated microsatellites can effectively predict the MSI samples. Well, this is a result of the validation using another cohort. We compared our 20 microsatellite markers with conventional markers, but microsatellite markers. And these, our microsatellites are highly correlated with the result of the BAT markers. So we also analyzed mutation in the MSI samples and MSS samples. We newly identified the mutation in APC gene. This is a very famous oncogene, and by the analysis of microsatellite. We also identified several important genes are mutated, has microsatellite encoding regions, and they are mutated. So we then analyzed the mutation in mismatch repair gene and proofreading genes, and the association with the MSI samples. As expected, these mismatch repair genes are highly mutated in MSI samples. So this is a summary of the first part of my talk. So we analyzed approximately 2.2 million microsatellites. And we identified highly mutated microsatellites, and we identified MSI samples, and we found the replication timing and DNA shape are significantly associated with mutation rate of microsatellite. And we also found that the 20 highly mutated microsatellites that can be a biomarker to detect MSI. So analysis of mutation in mismatch repair genes shows that somatic SNV and short indels have larger functional impact than germline mutation and structural variation. So this study was published in 2020. So I think this our software have been released from GitHub, so you can use our software to identify somatic mutation in microsatellite regions. So next, I would like to talk about the systematic analysis of mutation distribution in 3D protein structures. So driver gene is very important concept in cancer genome sequencing project. So driver genes is a gene that directly affects the carcinogenesis and the cancer development. But identification of driver gene is not a very simple method, simple problem. So in general, driver genes are detected by the number of mutations in each gene. The number of mutations are tested and compared with the expected number of mutations in each gene. Okay? And the highly mutated genes are identified as driver gene candidate. So the statistical test that analyze the number of somatic mutations is called gene burden test. Okay, gene burden test has identified driver genes. However, we consider that the analysis of new aspect of driver gene can identify novel driver genes. So we focused on the protein 3D structure. So this is a previous study, about a very old study. So this study identified DIS3 gene as a driver gene in multiple myeloma. So this study also shows that the somatic mutation of these two somatic mutations are very close in the protein 3D structure. So these two mutations, important mutations, are not close in the amino acid sequence. However, these two genes are very close in the 3D protein structure. So we consider that the analysis of 3D protein structure provide us with novel driver genes. So we developed a method to identify novel driver genes. We named this 3D permutation test. So 3D permutation test is very simple. 3D permutation test generate the distribution of the distance among mutations using a simulation. Okay, so if one gene has 3 mutations, so we randomly selected the 3 positions and calculated the distance among these mutations. So this process repeated 10,000 times or 1 million times, and the distribution of the 3D distribution of the distance of mutations are generated. And using this distribution, we can test the observed distance of the mutations. Using this method, we can test the distance among the somatic mutations. So we download the data from TCGA and remove the possible SNPs and perform apply the 3D permutation test. So we analyze the 21 cancers. So this is a QQ plot. So this is a list significantly associated genes. And in this figure, we showed that the genes detected by multiple cancer types. As you can see here, the TP53 is a top ranked. And in addition to the TP53, PS3 kinase, and P10 and CDKN2A, they're very famous genes are correctly identified by our method. But in addition to these very well-known oncogene and tumor suppressor genes, our analysis identified a novel driver gene candidate. So we then compared the proportion of the significantly associate significant genes and non-significant genes between the tumor suppressor gene and other genes. As you can see here, our driver gene candidate detected by a method have a significantly higher number of tumor suppressor genes and oncogenes. So this result strongly suggested that our method correctly identify the driver genes. So we also compared the average distance between mutations, a significant tumor suppressor gene, significant oncogenes, other novel significantly associated genes, and other genes. As you can see here, the distance of the mutations are quite significantly different between the tumor suppressor and oncogene and other genes. As you can see here, the oncogenes, the mutation in oncogenes are tightly clustered compared to the tumor suppressor genes. So this result strongly suggested that the distance among mutations provide us with a very important information to predict the function of the genes. So we performed the gene set enrichment analysis of the significantly associated genes. As you can see here, the pathway related to cancers are identified, and protein kinase, apoptosis, and cell death are genes related to these terms are significantly enriched in the driver gene candidate by the 3D permutation test. So this slide shows an example of the significantly associated genes identified by the 3D permutation method. As you can see here in LAC1, LAC1 is tumor suppressor genes. So the LAC1 had the mutation in the LAC1 is not close in the amino acid sequence, but very highly clustered in the 3D protein structures, TGF, beta R1, DICER, and FAS. And the mutations are not very close in the amino acid sequence, but tightly clustered in the 3D protein structure. So we consider that the application of this method would identify novel driver genes. Next, I would like to talk about the long-read sequencing technology. First, I would like to talk about whole genome sequencing using long-read sequencing technology. So we sequenced the 11 Japanese liver cancer and matched normal samples. So to compare with short-read sequencing technology, we selected the 11 Japanese liver cancer and matched normal samples that were previously analyzed in the International Cancer Genome Consortium. So in total, we sequenced 22 genomes. We sequenced them using Oxford nanopore sequencer. And we developed an analysis method to identify genetic variation, somatic mutation from a long-read sequencing technology. And this is an expected pattern of structural variation. It's a deletion, it's RNA insertions, and inversion, and chromosomal translocation. So we constructed the analysis pipeline named CANFAR. So this analysis pipeline performed mapping against the human reference genome and identify the breakpoint of structural variations. Then search the overlapping breakpoint, and this analysis pipeline filtered possible false positive structural variations. So we identified the about 70,000 insertion, deletions, and inversions intrachromosomal under 265 intrachromosomal translocations. We selected germline structural variations and analyzed the length distribution. X-axis indicates the length of insertion and deletions, and Y-axis indicates the number of insertion and deletions. As you can see here, the shorter insertion and deletions are predominant, However, there is a clear peak here in insertion and deletion. Then we identified the somatic structure variations. We analyzed the whole genome sequence of tumor and whole genome sequence normal and identified somatic mutation candidate. We performed the experimental validation of the structural variations. So B is blood and normal tissue, and C is cancer. So as you can see here, we can observe the cancer-specific applications. So this result strongly suggested that our method correctly identified the somatic mutations, somatic structural variations. So we compared the somatic mutations identified by our method with short-read sequencing technologies. This is a Venn diagram of the structural variations. So this is the number of somatic structural variations detected by short-read sequencing technology. And this is the number of structural variations detected by long-read sequencing technology. As you can see here, the long-read sequencing technology identified larger number of structural variations. So this result strongly suggests that the long-read sequencing technology can identify more number of structural variations. So we also analyzed the driver gene structural variation in driver gene candidate. So our analysis identified 4 previously unidentified structural variation in driver genes. This is a result of the structural variation in driver. This is an example of novel structural variation in driver genes. This is an example of TART, telomerase reverse transcriptase. This is an example of deletion in CDKN2A, a tumor suppressor gene. So we also identified the integration of hepatitis B virus in the host genome. So we compared the identified HBB integrations between short read and long read. As you can see here, the short read identified 25 HBB integration. However, the long read identified all integration and additionally identified 11 HBB integrations. So interestingly, HBB integration were found in centromeric regions. And sign, it's a transposon, a repeat sequence. So this result strongly suggested that HBB can integrate to repetitive regions. I think this may be an interesting finding. So we also estimated the mechanism of the insertion and deletion generations. But I think that this is very, very complicated, and today is time limited. I will skip this method. But anyway, we classified the insertion deletion into the deletion event and the insertion event. And we found that most of the germline insertions were caused by R insertion or line 1 insertions. And most of the long insertions are mediated by transposons. 91% can be explained by insertion of the transposons. But in the deletions, only 34% can be explained by transposons. So in addition to the transposons, our analysis identifies the insertion event of processed shield gene. Processed shield gene is a reverse transcribed insertion of the reverse transcribed messenger RNA. So this is the mapping result of the inserted sequences. The inserted sequences are mapped to another gene and mapped to the exonic regions. So long-read sequencing technology enables us to detect the processed shield genes. So we also estimated the cause of deletions. So, and I will skip this slide because this is today the time is limited. But we found that a lot of deletions are caused by non-allelic homologous recombination. So we compared this result with previous short-read sequencing technologies. And our analysis identified the deletions caused by non-allelic homologous recombination. So non-allelic homologous recombination is the mechanism of the error of recombination. And the deletion caused by non-allelic homologous recombination has homology in the breakpoint. So this type of structural variation was very difficult to be identified the previous short-read sequencing technology. So this result strongly suggested that our long-read sequencing technology newly identified deletion that's caused by recombination errors. So this is a summary of my the presentation of the third topic. So we performed the whole genome sequence using long-read sequencing technology for the 11 Japanese liver cancer matched normal samples. We correctly inferred events that cause insertion of deletions and found that the most insertions were caused by transposon and ARU is the most predominant source. So we also estimate the mechanism of deletions. So analysis of somatic mutation in liver cancer shows that a long read could detect larger number of structural variations than a previous short read study, and that the mechanism of cancer SV generation were different from that of germline deletion. So our analysis provides a comprehensive catalog of polymorphic and somatic SVs, as well as their possible causes. This result was previously published 3 years ago. And we applied our method to genetic disorders to solve the causative mutations. So we collaborated with clinicians and performed the whole genome sequence of 11 genetically unsolved patients using long-read sequencing technologies. So our method identified very big deletions in 2 patients, and our analysis solved 2 patients out of the 11 unsolved patients. So our analysis, we also analyzed the whole genome sequence of patients with very complex abnormal chromosome. So this is the chromosome G banding of chromosome structure of this patient. So this patient have 3 chromosome 21. And interestingly, the 3rd chromosome 21 is different from the normal chromosome 21. So the chromosome 21 is generated by fusion of long arm of normal chromosome 21. So we performed the whole genome sequence of this patient and estimated the true structure of this patient. And our master course student made effort to estimate the true structure of this chromosome 21, and we propose the model of the structure of this extra chromosome 21. And finally, I'd like to talk about the transcriptome sequencing using long-read sequencing technology. As I mentioned before, the short-read sequencing technology cannot observe the full-length transcript. However, the long-read sequencing technology enables us to directly observe the full-length transcript. So to analyze the full-length transcript from long-read sequence data, we constructed the analysis pipeline. So this analysis pipeline considers the alignment or error rate and possible alignment errors and artificial chimeric read generated during the library preparation process. Using this analysis pipeline, we analyzed the 42 pairs of HCC and matched non-cancer liver samples. So we analyzed the HBV and HCC-related liver cancers and performed long-read sequencing using Oxford nanopore sequencer. And we analyzed the data using a splice pipeline we generated before. Using the analysis pipeline, we identified about 60,000, 51,000 transcripts from HCC and matched non-cancerous livers. Of these, about 7 or 8% are novel transcript compared to a database. So we compared the expression level for each transcript and identified significantly differentiated transcripts. About 4,000 and 5,000 transcripts are significantly upregulated, downregulated in liver cancers. And we also identified a lot of driver genes are upregulated, significantly upregulated or downregulated in liver cancers. So we found a very interesting driver gene candidate. This is a driver gene candidate of L1 MET transcript. So in MET, famous driver genes, so one transcript was significantly upregulated in HCC. And this transcript, the exon 1, is originated from line 1, is a transposon. And only this transcript, the L1 MET transcript, was highly overexpressed in HCCs. So this result strongly suggested that the L1, the promoter within L1, may be activated in liver cancer and cause upregulation of this transcript. So we performed the overexpression experiment of the L1 MET transcript. The L1 MET transcript increases cell proliferation in 2 cell lines, and the L1 MET knockdown decreases the cell proliferation in 2 cell lines. So this result strongly suggested that this transposon fusion transcript is a very important driver genes. We also analyzed the expression from hepatitis B virus. So as you can see here, the in HCC and non-cancerous livers, HBB are expressed. And interestingly, this region, this is the structure of the HBB genome, and interestingly, only these regions are expressed from the HBB. So we found that a lot of transcripts are transposon related. So we identified the HBX human fusion transcript. And a lot of transcripts are transposon related. As you can see here, in the HBB and human transposon transcript are highly upregulated, highly expressed in liver samples, in liver cancer samples. So this is the structure of the HBB and transposon fusion transcript. And these regions are HBB, and these regions are human transcript. Several transposons are fused to the HBB transcript. So we performed the overexpression analysis of the overexpression experiment for these HBB human chimeric transcript. All human HBB transcript increase of the cell proliferation. So this result strongly suggested that these human transposon fused transcript plays an important role in carcinogenesis. So we also analyzed fusion genes. So our analysis correctly identified the fusion gene candidate. So and we performed a validation experiment using RT-PCR. So we compared fusion genes detected by a method with fusion genes previously identified by short read. As you can see here, long read identified a lot of novel fusion genes that are not detected by previous short read sequencing analysis. So this result strongly suggests that long read would identify novel fusion genes that are not previously detected by short-read method. This is a summary of the fourth part of my talk. So we develop a nice pipeline splice for the nanopore long-read sequencing technology. So we analyze the 42 cancer and mashed liver cancer samples and analyze the data using splice pipeline. So we identified more than 700 the differentiated transcript-specific genes. We identified that the L1 MET transcript are significantly upregulated in cancers. And we also identified a lot of HBB human non-coding fusion transcript. We identified a lot of fusion genes, and a lot of them have not been detected by previous short-read sequencing analysis. So these results strongly suggested that long-read sequencing technology allows us to analyze the full-length transcript and show the importance of the splicing variant in carcinogenesis. So this analysis, in this study, we analyzed the 42 cancer and mashed liver samples. But now we are sequencing additionally more than 100 liver cancer samples using long-read RNA sequencing technologies. And we are now identified a lot of novel driver gene candidate and fusion gene candidate, and we are now, and the functional analysis is ongoing. So this is the collaborators. So thank you very much. And I hope this presentation leads us to possible future collaborations. I really appreciate this opportunity to present my talk. Thank you very much.
So thank you very much, Professor Fujimoto, for your great talk. I think we learned a lot of your studies, and we are quite interested in learning. And as a collaborator, I'm happy to collaborate with you further regarding African genome in terms of cancer genome especially. And we've got a lot of questions, and hopefully we can finish this, but we go maybe one by one. So Maria Jose Luis, she asked us about the impact of your study on the therapy. So for example, if you think about the therapy, how your study can be connected.
Okay, thank you very much. So I think that the identification of novel driver genes is very important to find the target of personalized medicine. I think that a lot of mutations are still VUS, variant unknown significance, but that is a consideration of the 3D protein structure provide us with a very important information. So this study we performed 8 years ago, and at that time we could not use the 3D protein structure of all genes. So we analyzed the 3D protein structure obtained at that time. But now we can use prediction of the 3D protein structure by AlphaFold 2 method. AlphaFold is very provide us with a very accurate prediction of the 3D protein structures. So now we are analyzing the 3D protein structure obtained by AlphaFold 2 and the new mutation list obtained from TCGA. And we can identify the larger number of novel driver genes. The functional study is now ongoing. So I think this can provide us with the important information for the further clinical applications.
Thank you very much. And then I'm personally very interested in your method for this 3D structure analysis. And it would be very nice if we can get African cancer genome sequence and then try to collaborate with you in terms of the analysis. That would be really great. And using AlphaFold and other softwares. And another question coming from Emmanuel in Nigeria. I don't know if I'm pronouncing very well. He was curious about the technique that you use for short read. Is that Illumina?
Yeah, that's right.
Okay.
So we
And-
use Illumina short read sequencing technology.
Okay, and then he was also asking about the coverage and the depth of sequencing. But I think basically, if you go to your papers, like these informations are found.
Yeah, that's right. That's right.
All right. And then another thing, it's kind of a recommendation and suggestion. Yosa mentioned about MSI along with TMB and BDL1 expression are very important biomarkers and the scores that are needed by oncologists to decide if the patient can benefit from immunotherapy. So thank you very much, Yosa, for the recommendation and comment. And Professor Tim Chindia asked about RSA. So could you please specify the question, because I didn't completely understand what you asked us, if Tim is there.
Yeah, in the meantime, just Hiroki, I just want to first thank our guest for the excellent presentation. You very well showed the importance of making use of the genetic data that we can generate from sequencing, not just to identify mutations, but also to work on the functional validations of those variations, which is something that is extremely missing in Africa and North Africa. So we hope that we will build collaborations with you to work on this part, because it's extremely important in the field of drug discovery, and also to try to reclassify some variations. As you said, the variant of unknown significance, it's a huge challenge for precision medicine implementation and to test patients. So it's extremely important to work on this part of genomic study, functional genomic studies. I just have one question about the in vivo or in vitro assays that you can do after the 3G analysis. Have you been able to work on this part or not yet? And if yes, what kind of technologies or assays that you would recommend for this? Thank you.
Excuse me. So thank you very much for very good comment, a very important comment. So, and your question is about the experimental validation of the result of the 3D proteins. So we are not expert of molecular biologists, so we are just using knockdown of using siRNA, and we are now making expression vector using a with mutation, and make a stable expression cell line and test the effect of some drug, of anti-cancer drugs. So, but I think that in future we would like to use how to say the test multiple mutations simultaneously using next generation sequencing barcoding method. That's very important, but we are now using a very conventional method. So I think that I'd like to ask you to tell us a method of the experiment of molecular biology.
Yeah, thank you very much. This is what I can, maybe Hiroki is much more experiment, has.
Well, I mean, this gives a great opportunity to discuss all together how we work on, because I used to work and, plan to work with Professor Fujimoto, and I work with Yosa, so we can discuss about the function validation, how we should do it, and what is the decent method and so on. But I think this is a really good opportunity to put all the things on the table, and then we can discuss all what we can do.
Yeah, I see also that our colleague Ines Bini is with us. She's now in Switzerland, but she's with us now, and she will be working on this part of functional genomics, and she's putting in place a platform for functional genomics in Tunis. So I think that altogether with different other African and European and even Asian partners, we will be able to really improve the work that we are doing in genomics, and this has a very important impact on the implementation of precision medicine. I can also see that we have someone from Incaba Biotech. Thank you very much, William, for being with us. And William is suggesting the PacBio technology as well. You use the Oxford nanopore, but we also know that PacBio are doing great job for long reads sequencing. Yes.
Yeah, that's right. That's a very good question. So I think that we used nanopore because the nanopore is cheaper than PacBio. However, now the cost of the PacBio is reducing. And the PacBio is now slightly more expensive than nanopore, but I think that the costs are not quite different. So I recommend you. I think that now the PacBio is better for whole genome sequencing, but I think that for transcriptome sequencing, we don't need to base pair accuracy. So I think that for the transcriptome sequencing, I think also nanopore may be better.
Okay, great. Thank you.
So actually, like we don't find like team. So I think so many questions and so many curiosity and interest and everything. But the next speaker is waiting for us. So maybe now is the time to let the next speaker talk about the presentation. Dr. Daniel Oliver.
Yes, thank you. I am Maria Jose Ruiz. I will introduce our next speaker. It's a pleasure to introduce Daniel Olivier. Dr. Olivier is an independent researcher and consultant, formerly associated with the Department of Chemical Engineering and Polytechnic of Montreal, Canada. He received his PhD from the Stellenbosch University in South Africa in the 21. And he followed 2 years of postdoctoral training. Subsequently, he joined the Pathology Research Facility Laboratory in the Department of Pathology at Stellenbosch University. His current interesting focus on enhancing the personal medicine workflow for breast carcinoma in South Africa, aiming to streamline the existing pipeline and prepare the functional genomic operation for commercialization. I invite also you to read one of the last articles of incorporating functional genomics into the pathology supported genetic testing framework implemented in South Africa. And the floor is yours, Dr. Olivier.
Hi there. I just want to make sure that you can see my screen.
Can you share your screen? It's at the bottom.
Can you see it?
Yes, we can now. Thank you.
So you can see the title of my presentation.
Yes.
Perfect. Right. Hello, everyone, and thank you all for joining this webinar. So during the next 30 minutes, I will share some of the work our group has been conducting in South Africa. Specifically, I'll discuss how we intentionally yet unexpectedly ventured into the research area that is the focus of this webinar, which is the application of functional genomics in personalized medicine. And subsequently, I will highlight the value of functional genomics, supplying an overview of what functional genomics entails by discussing easily understandable examples, before discussing some of the models and experimental techniques that can be used for functional genomics. And towards the end of the webinar, I'll highlight important considerations and place our learnings in the context of resource-limited settings such as South Africa. So before we get the ball rolling, I'd like to point out that I'm associated with various departments within Stellenbosch University in South Africa in a supervisory capacity, but also a consultant for Genomics, which is the company-used pharmacogenomic IT system our group uses to carry out personalized medicine. So back in 2021, while I was doing my first postdoctoral fellowship, my mentor at the time called me into her office and said that she needed she had a colleague that needs help testing the effect of a genetic variant. And this then would eventually become the project that we do today on functional genomics. So I was tasked to determine the functional impact of a rare TP53 variant in a South African family that at the time was classified as a variant of unknown significance. The thing is, I was trained mostly in analytical biochemistry and just graduated with a PhD in physiology. So genetics wasn't my strongest suit, but I knew how to problem solve. I loved the area of breast cancer research and was very familiar with assays to test biological effects. And also at the time, the concept of functional genomics didn't really exist in my vocabulary. However, being familiar with the hallmarks of cancer, I designed a set of experiments to try and unravel the TP53 variant mystery. And these experiments are currently being carried out by one of my PhD students. And recently, this work allowed my PhD student to attend the 72nd Linda Nobel Laureate meeting in Germany. So I'm really super proud of the work that she's been doing. However, we've also presented some of the results at international conferences, including St. Gallen and the International Congress of Human Genetics. But why is it important to determine the functional impact of this rare TP53 variant? So chances are good that if you're attending this webinar, you are aware that the gene TP53 and its protein product P53 is considered the guardian of the genome and mainly functions as transcription factors. Physiologically, when wild-type P53 accumulates in cells, they promote the expression of cyclin-dependent kinase inhibitors, DNA repair proteins, and pro-apoptotic proteins. Additionally, wild-type P53 also inhibit the expression of oncoproteins. So functionally, wild-type P53 promotes cell cycle arrest, allowing for DNA repair, the failing of which leads to apoptosis or cellular senescence. However, loss of wild-type P53 function due to, for example, mutations have been linked to cells being insensitive to growth inhibitory signals, the development of limitless replicative potential, and also genomic instability. But research also suggests that one mutant in the TP53 allele and one intact wild-type allele can lead to dominant negative effects, where the mutant P53 protein binds to the wild-type P53 protein and thereby activate the function of the wild-type protein, or lead to a significant loss in function. Now, the variant that we're investigating, the one that kickstarted our functional genomic research, was identified in index patient in 2019. So in 2017, one of the oncologists that forms part of our research team requested extended genetic testing for a female patient that presented with invasive primary carcinoma at the ages of 38, 53, and 59. Now, based on the index patient's pre-screen questionnaire, the team identified that the patient had a family history of cancer. Specifically, two of her sisters had breast cancer before the ages of 46, one of which passed away of breast cancer at the age of 48. She also had a mother that was diagnosed with melanoma twice and was also diagnosed with lung cancer at around 80 years of age. Moreover, the index patient's maternal aunt and uncle had lung cancer and prostate cancer, respectively. However, upon discovering that the index patient's maternal cousin had been diagnosed with brain cancer, our genetic counselor made the connection that this could be a case of Livermany-like syndrome. So this suspicion then met the comfort criteria for TP53 genetic screening, leading to the identification of a rare variant of unknown pathogenicity. And this variant is then called the TP53N340D. So as part of our exclusion testing, we started by excluding BRCA1 and 2 genetic variants using founder genotyping and multiplex ligation-dependent probe amplification. Subsequently, we sequenced the exome of the index patient or sister and paternal cousin, which led to the identification of this variant of unknown pathogenicity on the maternal side of the family. And this result was then also validated with Sanger sequencing. Now, based on the information available at the time, Varsom predicted that the variant would be deleterious, since the variant was absent in case control studies of more than 100 individuals that we conducted. Additionally, the variant was reported only twice in a database of more than 80,000 tumors, while absent in population databases at the time. Finally, the variant forms part of TP53's beta isoforms, which has previously been implicated in breast cancer. So subsequently, we also used genome sequencing, splice variant prediction, and MLPA for TP53 and CHEK2 rearrangements to exclude other potential genetic causes. Now, key step that is not visible here is how the information was compiled and presented to the treating oncologist. In a lot of difficult medical cases, information can easily get buried in patient notes and files. As such, having a platform that can compile all results and create a short case report that the treating physician can use for medical decision-making is really crucial. So we use a pharmacogenomic ID system called Genomics, which have been extensively validated to the extent that in 2020, this system and the framework it uses won its creators the International Best Practice in Personalized Medicine Award from the International Consortium on Personalized Medicine. Now, in short, in this framework, patients are referred by their physicians to complete an ethics-approved medical questionnaire on a mobile application, which captures data on personal and family medical history, lifestyle factors, and past treatment responses. Based on the information, the patient is then tested for BRCA1 and 2 founder mutation, in addition to non-communicable disease variants useful in the differential diagnosis of lifestyle-related and/or inherited cancers. Additionally, tumor biopsy samples are screened using a variety of microarray and NGS technologies to inform treatment options. Depending then on the genetic findings, the patient is sent for a specific pathology test that could support the genetic findings and assist the treating physician to make informed decisions. If these tests are negative, the already available DNA sample is sent for exome sequencing, and depending on the outcome of the exome sequencing data, genome sequencing is employed for validation or as a more comprehensive screening tool. Now, during the entire process, genetic counselors facilitate the knowledge transfer to the patient to ensure that the patient is always well informed. However, the current workflow is not without its challenges. Amongst these are the frequent identification of variants of unknown significance that can significantly hamper the medical decision-making. So to overcome this challenge, we've invested in an additional wet lab functional genomic component to this framework, which is currently under development. However, in the future, I'd like this framework to first make use of artificial intelligence and large databases before moving to wet lab functional genomics. This could help speed up the variant classification by potentially, for example, combining prediction tool scores into Z-scores that will allow for a more objective assessment of whether a particular genetic variant is likely to be pathogenic or benign, or alternatively to look for sequencing patterns in patients' data that matches those of patients with an established diagnosis. So, considering the incorporation of functional genomics in our workflow, we recently reviewed the literature of how functional genomics have been implemented in the past, focusing here specifically on carcinomas. But what is the value of functional genomics? Or alternatively phrased, why functional genomics? So to illustrate the value of functional genomics, let's consider the following theoretical case. We have the challenge that we identify a TP53 variant with an unknown pathogenicity that is likely the cause of a patient presenting with multiple breast carcinomas. We know that the gene TP53 and its protein product P53 is a tumor suppressor, and having a pathogenic variant in this gene could lead to Liframeni-like features characterized by a predisposition to a range of cancers. So using functional genomics, the idea is to gather experimental data that will assist in variant classification or reclassification, which could alter treatment choices. For example, our findings could lead to the variant being reclassified as likely pathogenic or pathogenic, which could lead to treatment recommendations such as mastectomy and the avoidance of radiotherapy. Unfortunately, our evidence could also yield no additional information, meaning that our variant of unknown significance will stay a variant of unknown significance. But regardless of this fact, we would still gain important scientific information that can assist in revisiting our initial hypotheses. So what does functional genomics entail? In short, it's the selection and use of model system and experimental methods to link genotypic information to cellular responses or phenotypic outcome. For example, let's use the previous case on the previous slide. We know the impact that a pathogenic TP53 variant could have for individuals carrying one, but we don't necessarily know the impact of this variant. So the idea is to select an appropriate model and experimental method, for example, as shown here, to collect data that will serve as evidence to support pathogenicity or lead to the reclassification of benign. So hypothetically, let's say we detect a variant of unknown significance in the DNA binding domain of P53, which is where approximately 80% of TP53 variants occur. So considering that P53 expression is associated with various stress cues such as DNA damage, hypoxia, and oxidative stress, the idea is to recreate one of these conditions experimentally and then measure the cellular response or phenotypic change. Since a mutation in the DNA binding domain of P53 could affect the expression of downstream genes, such as the failing of apoptotic signaling, the hypothesis then is that we'd be able to measure this cellular response experimentally. So in essence, we select a model such as cell culture and grow, for example, MCF12 breast cancer epithelial cells. Then we induce the variant we're interested in some cells while keeping others wild type. Subsequently, we treat the cells with a stressor, such as, for example, doxorubicin, that induces DNA damage, forcing P53 to respond. Finally, to measure the cellular response, we can use metabolic activity assays, for example, to measure cell survival. If the viability of the cells containing the variant shows increased cell survival, the variant likely affects cell death pathways such as apoptosis and is likely to be pathogenic. So to concretize this idea, let's consider the variant we're working on. So due to various factors and considerations, we opted to use peripheral blood mononuclear cells as our model. Herein, we treated PBMCs from individuals carrying no pathogenic TP53 variant and individuals carrying the M340D variant with doxorubicin to induce P53 signaling. Whereafter we then measured cell survival using a metabolic activity assay. And here are some of the preliminary results, which is currently grouped by individuals that have breast cancer and those that don't. So as you can see, there's an increased tendency for PBMC cell survival in individuals carrying this specific variant. Therefore, it's likely that this variant could affect cell death signaling pathways. However, given the complexity of this gene and where this variant is located, it is too early to definitively conclude that the variant functionally affects P53 signaling. Unfortunately, selecting the right model in assays like these can be challenging, to say the least. So when we look at various model systems, we find that, for example, in vitro models, they are simple to conduct, reproducible, cost-effective and can be implemented at a high throughput pace. And you can also control for a lot of the environmental variables. However, these assays fall short in representing physiological complexity, and they don't always yield clinically relevant outcomes. In contrast, in vivo models represent physiological complexity really well. However, these models are sometimes unsuitable for high throughput analysis and do not always directly translate to clinical responses in humans due to inherent genetic differences between species. In contrast to these methods, ex vivo models probably represents, most accurately represents the organismal phenotype, but low general population frequency of rare variants limits the number of individuals that can be available for research purposes. Additionally, these models cannot fully mimic intracellular communication, physiological processes, and disease complexities within the human body. So there's a lot of experimental methods that can be employed to assess changes. And in this webinar, I will highlight a select few key approaches, but please note that this is not an exhaustive summary of all the available methods. Moreover, it's important to realize that the methods will largely depend on the disease being studied and the gene under investigation. So first up, we have, for example, protein modeling and structural bioinformatic analysis, which can offer valuable information for predicting the classification of variants of unknown significance by assessing its impact on protein structure, stability, and molecular interactions. Here, for example, homology modeling, molecular docking, molecular dynamic simulation, and residue interaction network analysis are some of the techniques often employed to model and predict the impact of genetic variants. Combined with variant effect prediction tools such as CAD, Polyphen2, and VEP, protein modeling tools offer a cost-effective first-tier solution to investigate the impact of genetic variants. Equally cost-effective is database mining and machine learning. For example, large databases such as the Human Genome Mutation Database and ClinVar catalog genetic variants and associated disease and drug response phenotypes. As such, these databases offer a rich source for information. Unfortunately, rare variants are often absent from these databases or have very limited information. Luckily, tools like CAD, Polyphen2, and Revel, which utilize machine learning trained on known pathogenic, known mutation functional effect relationships from various databases can predict the impact of new mutations on protein function or cellular pathways. In contrast, multi-omics, which include transcriptomics, proteomics, metabolomics, and epigenomics, offer a comprehensive analysis of cellular changes on a genomic, RNA, protein, and metabolite level. While these methods are comprehensive, you pay a steep price for including them in your pipeline, not only from a consumable point of view, but also in terms of the level of skill required to make sense of the data and the time needed to draw good conclusions. As such, simpler methods such as flow cytometry, metabolic activity assays, migration and invasion assays, and for example, tube formation assays allow relatively rapid downstream analysis of cellular responses. It's also less expensive consumable-wise, but also in terms of the level of skill required for researchers and the time needed for data analysis. So at our facilities currently, we can look at one variant at a time. However, a more comprehensive and high throughput approach is the use of multiplex assays of variant effects. So these assays, which include massively parallel reporter assays, deep mutational scanning, and saturation genome editing, can assess thousands of variants simultaneously. And at the current rate of variant discovery, as more exomes and genomes are being sequenced daily, MAVs offer a practical solution to overcome the investigation of single variants. Moreover, the aggregation of all individual MAV datasets can assist machine learning models to better classify variants. The question, however, is to what end do we perform functional studies? Should we publish the functional data as is, or is there a better approach? So unless your results are placed within the framework for interpreting and reporting sequence variants to ensure consistency, accuracy, and clinical utility in genetic diagnostics, your work might not immediately be useful for the larger scientific community if it's published as is. A better way is to place your findings in the context of expertly curated frameworks, such as those from the American College of Medical Genetics and Genomics, and the Association for Molecular Pathology. Now, these standards and guidelines work by outlining a systematic approach to evaluate genetic variants based on evidence categories and criteria. So by assigning points to genetic variants based on multiple evidence categories, such as population data, computational predictions, functional studies, and segregation analysis, these standards and guidelines allow you to classify your variant of interest into categories like pathogenic or benign with more rigor and confidence. In turn, I argue that doing so could significantly impact the relevance of your work, which might even make it more attractive to higher impact journals. So, in summary, our recent review highlighted the various model systems and experimental methods for functional genomics in the context of breast carcinomas and their strengths and weaknesses. Retrospectively, this work also highlighted a couple of missed opportunities to me. Specifically, the published ACMG and AMG standards and guidelines, along with subsequent work by researchers like Brennig, offer valuable insight on how to approach functional genomic functional genomic studies. Had I known about these resources earlier, I would have designed our initial experiments probably a little bit differently, following their guidance from the start. Moreover, in resource-limited settings, a more effective approach to functional genomics might have been to first explore advanced computational methods that use machine learning. This would then have been much more cost-effective than starting out with wet lab experiments. Furthermore, having worked with PBMCs as our model system to resolve the functional impact of our TP3 variant, germline variant, I can tell you that they are not necessarily the best model system. These cells have several drawbacks, most notably being their limited replicative potential and the variability of PBMC isolates between patients, which affects standardization. Our review of the literature indicated that a better approach might have been to first create induced pluripotent stem cells, which can be routinely cultured before being differentiated into cell types that reflect the origin of the disease or the cell type that would be affected by the variant. However, I am very aware of the drawbacks of iPSCs, primarily the time it takes to establish and validate iPSC colonies. But in the context of shorter timeframes to the return of research results, this might be a significant limitation. But with current automation systems, I think that iPSCs still offer a better solution than PBMCs. But regardless of the drawbacks of PBMCs, we've been working on setting up assays and standard operating procedures that can be used for functional studies. For example, flow cytometry, metabolic assays, metabolic activity assays, and migration and invasion assays, but also some other methods like Western blot and fluorescent microscopy, which is not shown here. So to place some of the learnings we have from the past 3 years into perspective, I have a couple of recommendations for functional genomics in resource-limited settings such as South Africa. The first being, before attempting functional genomics, thoroughly explore guidelines such as those set out by the ACMG and AMG and related literature. These resources will ensure that you are well informed to make decisions related to functional genomics and how to approach the models and experimental methods needed to reclassify variants. In turn, this knowledge will prevent wasting valuable resources. Second, once you grasp the guidelines, as a first experimental step after genetic sequencing, prioritizing techniques such as protein modeling, structural bioinformatics, database mining, and machine learning is my second recommendation. In this regard, platforms such as the National Institute of Theoretical and Computational Sciences in South Africa, which are dedicated to advancing theoretical and computational sciences, is ideal for capacity building and learning. Third, should the evidence gained from step 2 suggest a variant to be detrimental or likely detrimental, validate your computational findings with wet lab experiments, drawing on your knowledge of the disease, the gene of interest, and how the variant could contribute to pathology. Lastly, while ex vivo models may better reflect the organismal phenotype and certain experimental methods may provide more comprehensive data, do not overlook the value of simpler models and methods. Ultimately, the sensitivity and specificity of your model and experimental method are what truly matters. But I have been asked to list a couple of areas for potential collaboration. So first off is collaboration with the medical community to maximize the cost effectiveness of our research. So obviously, the more people that makes use of our services will significantly affect how much our services cost, for example, exome sequencing. Secondly, partnerships with research groups specializing in computation that can deliver results quickly following the discovery of variants of unknown significance. And then third, partnerships that could bring expertise and funding assistance to, for example, establish a functional genomic research center in South Africa that will enable researchers to conduct their studies or request the assessment of their variants. And then lastly, forming a network of experts who can assist in variant classification and ensure that we comply with guidelines such as those set out, for example, by the ACMG and AMP. Lastly, I'd like to acknowledge the following people who have been instrumental in some of the research that we've conducted. And I'd also like to acknowledge our hosts for inviting us to this webinar. We really appreciate the opportunity to present our results. Thank you.
Thank you very much, Dr. Olivier, for your presentation on the pathology-supported genetic test for breast cancer in South Africa. You highlight the importance of incorporating functional genomics to address the bottlenecks caused by variants of uncertain significance, and you give also given the variety of model system and experimental methods available, you have also elaborated a recommendation on how to prioritize which functional genomic technique we can employ. Thank you very much. But additionally, I was thinking, what are the main challenges you face in integrating these methodologies in the existing framework? And mainly how you propose to overcoming them?
Thank you for the question. It's actually a very loaded question. I think one of the main challenges we currently face is in interpreting genetic findings and functional results, specifically in terms of classifying variants based on how quickly different guidelines actually evolve. For example, things can easily change from a variant being considered pathogenic with older guidelines, while newer guidelines might actually classify the variant as maybe for a variant of unknown significance or benign. So that's probably one of the biggest challenges we have, is that as quickly as guidelines change, you're not always sure, would a variant stay pathogenic or would it actually change to something else?
Thank you very much. Yours, you have a question?
Yes, thank you very much, Maria Jose, and thank you a lot, Dr. Daniel Olivier, for being with us today. I know that it's around 6 or 7 a.m. in Montreal, so thank you very much for being with us today. It's an excellent presentation, and I think that your talk completed the first talk, which was more on in silico analysis, and you presented very well the experimental part that can complete the functional genomics aspect. So my question was about variant of unknown significance. First, it's not a question more, it's a comment. So we are putting in place a kind of collaboration with the Enigma consortium. The Enigma that are really classifying a variant of unknown significance in different populations in the world. And it's a huge consortium that includes different countries, including Canada and other European and African and North American countries. So I think that it would be a good way to proceed for collaboration between all these consortia to try to assess and interpret the genomic results. As you said, it's a very huge challenge. So I think that by working together, it will be a good way to try to reclassify some of those variants. This is my first comment. Now the question that I have, you are focusing on variant of unknown significance. And as you said, you are more like referring to the ACMG guidelines. But what about novel mutations that we can discover in African populations? I think that this is also important to focus on this type of variants because, you know, we are sequencing, we are identifying lots of novel mutations from Africa. And I think that we also need to work on these variations to validate their functional impact. So what do you think about this?
No, I completely agree.
Okay, and it's the same approach that you are proposing for novel mutations, or it's something that could be a bit different? In terms of infrastructure, experiments, I think that should be the same.
Well, Hiroaki Taniguchi from Polish Academy of Science and also UM60 Morocco. I'd say that the best is like we do all the function validation, but as Olivier said, it takes sometimes too much, like if you do like still in vivo experiments. So I think we have to know the factors and the mutations. So for example, if you work on transcription factors, so you know the role of the transcription factor is like to activate the target element. So it can be done to know the impact of the pathogenesis, like by doing simple luciferase assay, for example. And if it's epigenetic factor, you can do like chromatin immunoprecipitation type of assay to know the histone modification capacity and so on. So before going all the things, like including invasion and cell growth and all that, I think we can use like, for example, in silico approach and also like simple assays, which you can do it within 2 to 3 weeks. And to get the first screening done and then choose the hit and do more. For example, CRISPR-mediated gene editing and all that, because all the process would take a long time, and you don't want to find out that, okay, nothing happened after you make all the effort. So this is like kind of the message from my side, and I'm trying to do this way all the experiment. And also for the collaboration opportunity, I'm situated in Morocco right now, so if we can collaborate together, this will be also nice. So this is like kind of my recommendation based on the experience that I have. And thank you very much, Olivier. I totally agree with your opinion and the presentation and so on. Thank you.
Thank you. You highlighted a really good point there. It's always a challenge finding these variants, and getting a result to the patient is not always a quick process, especially when variants could be in areas of the genome that's difficult to assess. So I would definitely say being able to have functional assays that can be implemented at a relatively quick pace, where the return of research results, as you said, I think 2 to 3 weeks is definitely the preferred option, and definitely a good choice.
Thank you.
Thank you. There is a last question from Marwa. Madhuani. That is, what do you think about functional laboratory testing?
Can you please repeat the question?
What do you think about functional laboratory testing? I don't know if Marva, would you like to explain your question more?
I think that she's asking about what do you think about implementing different functional laboratory testing in Africa? So for functional validation of different variations. Because as I said, we don't have enough resources and we don't have enough laboratories that are specialized in functional genomics in Africa.
Yeah, I think what I would suggest, having good, reliable assays that could reliably interpret genetic findings, that would be the first class thing I would recommend. Not necessarily going into very complex methods. That is one, extremely expensive. So I would recommend setting up assays that could be reliable and very sensitive to genetic variants in classifying them. So having assays that is reliable is definitely one of the recommendations that I would make.
Yes, thank you. And I think that it's important to start by choosing the variants that have an impact for the clinical management of patients. So as you said, for the purpose of precision medicine implementation, it's important to start with, with variants that are known and they have a kind of conflicting interpretation of their pathogenicity. So this is the first step. And in parallel or at the second level, we can also try to work at the research field on the functional validation of novel mutations that come from African population. This is a good way to proceed, I think, yeah.
I think also what would be good is prioritizing genes that is mostly associated, for example, with cancer. In our case, for example, being able to prioritize genes that is a lot of times associated with specific diseases, especially diseases that that is frequently detected. Prioritizing those genes would be a good start.
Thank you very much. I cannot see another question. Then I think we are near to 2 here in Rome. And then maybe we can close the webinar. Yours, you can close.
Yes. First, I would like to thank the EU Africa PERMED for organizing this webinar with us. Thank you very much, Maria Jose and Erica, and all the team for all your efforts. And we hope that this collaboration between PERMEDINA and EU Africa PERMED will continue even after the end of these 2 projects, because they will end soon. And we are also trying to discuss about the next and the sustainability of these efforts, because it's extremely important. Also, I would like to thank our speakers. Really, it was extremely interesting what you presented, and it gives us an idea about how we can proceed in different African institutions and countries to really make use of the genomics data that we are generating. Functional genomics is a very, very important field, and we need to make efforts to work on this all together. And last but not least, I would like to thank the moderators, my colleague, Dr. Hiroki Taniguchi, and you, Maria Jose, for your time and for being with us. I would like to thank all the participants. Jihan, thank you very much for organizing all this and for your efforts. And we hope that we will have the opportunity to organize other webinars jointly with the EU Africa PERMED and different other consortia and initiatives in Africa. And I see that Maria Jose already put an announcement for the next webinar. If you can maybe just give a few words on that, because it's really important. And thank you all for being with us today. Thank you very much.
Thank you, thank you. Of course, we will continue our collaboration also in the new partnership on personal medicine.
Yeah. So the next webinar, Maria Jose, it will be on genetic testing, counseling.
Yes, genetic counsel, yes, with the African Genetic Association and also with the colleagues from Tanzania.
Great. Okay, thank you very much. Thank you all, and see you soon. Thank you. Bye-bye.
Bye-bye.
Before leaving, we have to do a group photo. So please, dear participants and speakers, if you can turn on your camera to do a group photo, to post it later on social media, our social media, and also social media of Africa Permed. Please, just a few seconds. I'm seeing some camera. Okay. For persons who can open the camera, sure. Okay, so I'm getting a photo now. Just another one. Smile, big smile. It's okay. Done. Thank you very much. Thank you.
Bye-bye.
Thank you all. Bye-bye.
Bye-bye.
So I thank you.
Au revoir.
Bye.